Good morning. Today I would like to present my paper entitled Detection of Rice Plant Diseases Using Convolutional Neural Network. We are a team of three from the Informatics Department, Faculty of Computer Science, UPN Veteran Jawa Timur. For introduction, in Indonesia, rice plants has been playing an essential role in Indonesia's economic life with the average rice consumption around 90.1 kilogram per person per year. So yes, we eat a lot of rice. Rice plants may also be infected with diseases, unfortunately, caused by weather, soil environment problem, viruses, fungus, other type of pests. So the accuracy of the diagnosis of rice disease is one of the key factors to determine the best treatment for rice disease. In this experiment, the convolutional neural network is a deep learning method with neural network approach commonly used to analyze visual imagery. Therefore, this research investigates detection of rice plant diseases from leaf images to determine a class among four types of diseases using a convolutional neural network. In general, a CNN architecture comprises of several layers. From the input layer, we extract the features into convolution layers, and then from feature maps, we extract more subsampling from the feature maps, and then another feature maps uh, in another layer of convolution. At the end of the layers exist the fully connected layers to generate the output, the final output. CNN applications has been used in many areas, such as for image recognition, recognizing or detecting images, objects in images. Also, it can be used as a recommender system, giving recommendation of place, people, things, etc. Also, it might be used in natural language processing, where CNN learn how to uh, determine and acknowledge the human language. In this experiment, the materials and methods are as follows. We use data sets of around 2,000 rice leaf images, which is split into training and test sets. So these are the examples of some images with four kinds of diseases, which is leaf blast, brown spot, bacterial leaf blight, and tungro. We use two convolutional layers with two subsampling layers and four fully connected layers. So here's our architecture. From input, we convert it first to grayscale and resize to 50 by 50 pixels, and then supplied into the uh, convolution layers. We use a ReLU activation function for the convolution layer, and the fully connected layers we use a softmax function. Results: all experiments were implemented in Python using TensorFlow class library. There are six uh, models that we generated while maintaining fixed values for number of epochs, batch size, and validation split. Epoch is 10, batch size is 32, and validation split is 0.1. So here's the result. We can see that the highest training accuracy is achieved with model ASA8. Also, it has the lowest training loss. So we use ASA8 to uh, predict the data sets. So these are the graphic plots of the accuracy and model loss from ASA8 model. From that model, we can predict all the test sets into four types of classes. The highest precision is reached in Tungro disease, Also, the recall is achieved in brown spot in recognizing the brown spot, while the overall accuracy is 51%. So from table 2, we can see that the system performed the best in predicting tumor diseases and the worst in blight disease, showed by the harmonic F measure values of 75% and 25%. The low accuracy is mostly caused by the wrong prediction of blight disease. So lots of blight diseases was mistakenly classified as a brown spot, probably because of the similarity of the images. However, the system was very good in recalling brown spot diseases with a high recall rate here and quite good at predicting tumor disease that indicates that it has successfully learned at least the two types of diseases. Uh, we know that there is no single best architecture applied for all kinds of uh, problems. So including how many layers and nodes, what types of activation function to use, etc. So this experiment uh, could be escalated by experimenting on more varieties of architectures, such as modifying the number of hidden layers and nodes in each layer. One approach is to use a genetic algorithm to select the best architecture model uh, on a specified case. As a conclusion, a CNN architecture has been proposed to predict diseases in rice among four types of disease: blast, blight, brown spot, and tumor. The best model generated by the system has this is the size of the nodes in each of the convolution layer 128. We use two convolution layers and two fully connected layers with the size of 16 and four final nodes. It has 91.41% in training accuracy and an average of 51.2% in testing accuracy. That's the end of my presentation. Thank you.